Hello, I'm Dr. Alex Murphy, and I work for Roche Pharmaceuticals as a medical director. And I'm here today to talk to you about micro-dystrophin and gene therapy. This second slide is just our disclaimer, and it's saying that we developed this presentation jointly between Roche and Srpta pharmaceutical companies, and it was finally approved by Roche. This is all part of our partnership between Roche and Srpta. So, neuroscience is a major part of research and development at Roche. Our goal is to pursue groundbreaking science to develop new therapies to improve the lives of people living with chronic neurological diseases. Roche's aspiration in neuroscience and rare disease is to preserve the innate brilliance inside us all, what makes us unique as individuals, beyond any disease or condition. It is this aspiration and commitment to develop transformational therapies that has led to Roche investing in gene therapy for rare disease. In December of 2019, Roche partnered with Srpta Therapeutics to collaborate to improve the lives of people living with Duchenne. Together, we will reach more patients with Duchenne faster than we could on our own. In order to talk about our trials, it's important to first understand what gene therapy is. Gene therapy focuses on the transfer of genetic material to a person to treat a genetic disorder. The aim of transferring genetic material is to compensate for genes that are not working correctly, or that could work better, or to make a beneficial protein. Gene therapy aims to promote sustained expression of the therapeutic gene, or as we call it, the transgene, at a level high enough to be considered therapeutic and make a difference to the patients. Clinical investigation using gene therapy first started way back in 1990, and now more than 2,500 clinical studies have been initiated for a broad range of rare and neurological disorders. In order for the potential of gene therapy to be realized in any therapeutic intervention, three essential components are necessary: the vector, the promoter, and the transgene. The vector refers to the adeno-associated virus, or AAV, that's used to carry and deliver the transgene to relevant tissues. The vector is also the main component that interacts with the host immune system, and thus is believed to be the major driver of the safety profile for the therapy. The promoter controls which cells express the transgene delivered by the vector. The promoter also controls how much transgene the target cells will produce. In the case of Duchenne, the aim is to get them into muscle cells. The transgene is, of course, a critical component of the whole thing. It intends to produce a functioning version of the protein of interest. For Duchenne, that would be dystrophin. Dystrophin itself is one of the largest genes known to man, and it doesn't fit within a viral vector. Therefore, micro-dystrophin has been developed and engineered so that it includes the most critical parts of the dystrophin while still fitting within the vector. When evaluating any gene therapy as a potential treatment for a genetic disorder, we must ask ourselves several questions, and these are evaluated in clinical trials. Firstly, what is the safety and tolerability of the treatment? And that's the same for any medicine. It's important to establish that first. Secondly, is the transgene in the target cells? Is the desired protein being made? And is it in the right place? These are all assessed through clinical trials using Western blot and laboratory techniques such as that. Finally, do patients show an improvement in function? And this is measured by various assessments, including physical examinations usually led by a physiotherapist. And these include assessments such as the North Star assessment. So this brings us on to the SRP-9001, which is the medicine in question. The first trial that was looking at this study drug was called Study 101. It's an open-label, single-dose trial. This means all participants received the SRP-9001. This was a phase 1 or 2A trial, meaning it was an early phase trial, that enrolled 4 subjects with Duchenne at the age of 4 to 7 years. The primary objective of the study was to evaluate safety and tolerability of SRP-9001. All patients were negative of the antibodies to the viral vector. All patients were on a stable dose of corticosteroids for at least 3 months prior to gene transfer. The second trial seen here is Study 102. It's a double-blind, randomized, placebo-controlled phase 2 study. This means that neither the patient nor the investigator knew whether they were actually receiving the SRP-9001 or a placebo, which is a medicine not expected to have any effect such as saline. 41 patients were enrolled. All are dosed and are in the follow-up period. Crossover has now started while still in the double-blind phase, meaning patients initially in the placebo arm are now being dosed with SRP-9001. Future trials support our goal to expand our eligibility criteria across different studies, with the first slated to begin soon. Our next studies include trials 103, which is a phase 1 trial, and a phase 3 trial, with age criteria similar to our studies of 101 and 102. In addition, we're currently planning a trial which will involve individuals who are non-ambulatory. It's important to note that with some medicines, as we develop them, small amounts are at first made using a particular method. When you want to make larger amounts of the medicine and scale up, it's important to show that the new way of making the medicine, called the commercial material, results in the same effect, and that the scaling up process hasn't meant it's less safe or effective. Study 103, which is now called Endeavour, is an open-label systemic gene delivery study evaluating the safety of and expression of SRP-9001 in 10 subjects with Duchenne muscular dystrophy at 12 weeks post-infusion, using this commercial process material. It's considered a phase 1 study and is thought of as a bridging study to assess if we get the same outcome with the commercial supply as we did with the clinical supply, the smaller volume, in our previous SRP-9001 studies, which is 101 and 102 I've just spoken about. This is separate from our global placebo-controlled phase 3 trial. Finally, we would like to thank the patients and their families who take part in clinical trials, as well as patient advocacy groups and caregivers who provide valuable input into the design of clinical trials and ongoing research. Thank you very much for inviting me to speak today, and please enjoy the rest of the conference.